The last type of muscle tissue is a smooth muscle that's located mostly in the walls of the hollow organs such as stomach, intestine, urinary bladder, uterus, and other hollow organs in the body. It includes spindle-shaped cells with central nuclei, just one nucleus. No striation. It means it don't have any sarcomeric structures, and the cells arranged closely to form the sheath of the muscle. They are spindle-shaped with flat nucleus in the wide part of the cells. They located in the internal organs, and they have involuntary control. Smooth muscle tissue have spindle-shaped cells schematically, and here with light microscope and H&D staining. They are single cells and central nucleus that located in the wide part of the cell. And when we, for example, prepare a section from here or here include the nucleus, and here don't include the nucleus. The narrow part don't have nucleus. Because of it, in the transverse section, some cells have round nucleus and some cells don't have nucleus. The cells are not striated. Why? Because no sarcomers. They include the actin and myosin, more actin than myosin. Why? They don't arrange as sarcomeric structure. They join together in other structures that I explain for you in the next slide. The fibers are smaller compared to the skeletal muscle. Under electron microscope, the smooth muscle have a central nucleus, mitochondria, yes, polyribosomes, and cisterna of the RER, and also SER or sarcoplasmic reticulum, Golgi apparatus, and also some pinocytic vesicles that transfer some materials from the outside of the cell to inside by exocytosis. In the smooth muscle cell, the bundles of the thin and thick myofibril crisscross obliquely through the cell and forming a lattice-like network. Yes, they form a network inside of the cell that these fibrils, thin and thick myofilaments, join to some structures that named dense body that located in the sarcoplasm, that because the dense body is a location for the binding of the thin and thick filament, some scientists believe that dense bodies instead of Z-disc contain alpha-actinin that join the actin, bind the actin to these bodies. In the smooth muscle, also actin and myosin contract by a sliding filament mechanism. But in the dense body, additional to the alpha-actinin, two other proteins that are intermediate filaments as desmin and vimentin located in the dense body, that they are a type of intermediate filaments that you know about them in the cells. The desmin located in the smooth muscle, vimentin also located in the vascular smooth muscle. As you see here, the thin filaments or alpha or actin filaments bind to the dense body by alpha-actinin, and because the thin and thick filaments form a network, a lattice-like network inside of the cell, when they slide together, the length of the cell decreased and contraction occurred in the smooth muscle. Now, thus you know the smooth muscle cell exception are no striation, spindle-like shape, and also they include the intermediate filaments such as desmin and vimentin. In the smooth muscle, there is no T-tubules. In the smooth muscle, or the calcium ion stored in the endoplasmic reticulum as similar to the skeletal and cardiac muscle. But for the involving this ion in the contraction, this ion should join to one protein that named calmodulin. Calmodulin and calcium complex activate the myosin light chain kinase, phosphorylate it, and some cascade occurred inside of the cell, and finally contraction occurred in the cell by sliding of the myosin and actin together. As you see here, calcium ion joined to the calmodulin. In the skeletal and cardiac muscle, calcium bind to the troponin. That is another exception in the smooth muscle compared to skeletal and cardiac muscle. About muscle regeneration, as I said in the first part of this session, there are some myoblasts like non-fused satellite cells that they act as a stem cell. They are inactive and they are reserve myoblasts. When the muscle injured, these satellite cells differentiate to myoblast and finally myocyte. But the skeletal muscle show very limited regenerative ability because the few number of the satellite cells. And the cardiac muscles lack satellite cells and amitotic cells. Because of it, they don't have any regenerative ability. For example, in the myocardial infarction, when the muscle injured and lost their function because they don't have any satellite cell, they enable to do regeneration and repair. But the smooth muscle have good regenerative ability. Why? Because they include a high amount of the satellite cells and mitotic cells. That these satellite cells have asymmetric division. Some of them differentiate to myoblast. That myoblast may be fusion in the skeletal muscle or not fuse in the smooth muscle. And some of them remain as a pool of the satellite cells and stem cell in muscle tissue. Briefly, muscle tissue is a soft tissue that composes the muscle of the animal bodies and gives to muscles to contract and movement occurred in them. In the mammals, there are three types of muscle tissue: skeletal or striated muscle, smooth or non-striated muscle, and cardiac muscle, which is sometimes known as semi-striated muscle. Smooth and cardiac muscle contract involuntarily, but the skeletal muscle contract voluntarily. The muscle tissue activate both by interaction of central nervous system and also as well as receiving the innervation from peripheral nervous system such as peripheral nerves or plexus, or endocrine or hormonal activation. The different muscle types have very different response to neurotransmitter and endocrine substance such as acetylcholine in the skeletal muscle, noradrenaline and adrenaline in the cardiac muscle, and nitric oxide in the smooth muscle. Also, the muscle tissue have some categories that depending the content of the myoglobin, mitochondria, and myosin ATPase activity that you know more about them in the biochemistry and also physiology. You can study this session in the Jankuira's basic histology in the chapter 10. Bye.